My name is Ulrich Günther. I'm here a professor of metabolomics at the University of Lübeck in the Department of Chemistry and Metabolomics. I started here 2 years ago. So my research has always been in the context of NMR. I have not always worked with metabolomics, but today it is mainly metabolomics in a biomedical context, and that includes projects more recently around COVID, but we also have cancer-related metabolomics, also measurements of metabolism in cell-based systems, liver-based projects, and a big part of my lab is now nutromics. We look at food products, mainly to discover food fraud. So I'm currently working on various aspects of metabolomics, but with the pandemic, COVID research has become very important and, to be honest, also provided new opportunities because we suddenly realized that a metabolic profile is highly relevant to diagnose the disease, and so COVID has become very important. Other fields in my lab are cancer metabolomics, more driven towards mechanisms of metabolism with flux analyses. We also work in foodomics, looking at food fraud, mainly in spices. These are the main projects in my lab. So the fascination about metabolomics is that you have something very applied. You are very close to the clinic. Your research becomes a lot more relevant than other things that we scientists do enjoy. You provide results that do have a true potential for diagnostics, also to understand disease mechanisms. If you look, for example, in cancer cells, this is the challenge of our time, to make science relevant for medical applications, and this is what I truly enjoy. When it comes to the advantages of NMR, you can ask my students. They will tell you that NMR is much more reproducible than mass spec. Mass spec is more sensitive, but the higher reproducibility of NMR is a huge advantage. You can do a measurement in Lübeck, and you can do the same measurement somewhere else in the world, and you get exactly the same result. This is not easily achievable with mass spectrometry. On top, you see lipoproteins and glycoproteins, which give you additional parameters with huge potential, and we're just about to learn how to use those efficiently. So I enjoy very much being part of the International NMR COVID network because it gives the opportunity to virtually and practically collaborate with colleagues around the world who are top-notch at the forefront of science. This is the advantage, and Bruker has organized that very nicely. We enjoy this. So our goal in the network is, of course, to provide relevant results. We've just done a lot of developments about glycoproteins. There's key expertise here in the group of Professor Thomas Peters in Lübeck, which we combine with our metabolomics expertise. We really hope that we can provide some additional solutions here where we can assign the glycoproteins and the glycan structures that we observe in blood spectra. We also hope that we can use our cohorts efficiently to provide answers for patients. Especially looking at long COVID, where we have a specific sample collection available, and our collaboration with this lipid apheresis clinic, where we get unique samples upon treatment of long COVID syndrome. We are investigating all kinds of COVID samples, which we get from very different sources. We have samples from our local hospital, intensive care unit. We also have a collection on campus where volunteers were asked in, about 3,000 people were asked in over several months to look at their COVID status, and about 50 have become corona positive. So we have a unique setup of samples where we can see before and after. On top, we are working with clinicians who provide treatment for long COVID. So there's one, Dr. Jäger, who I actually got to know through an article in the Frankfurter Allgemeine. And I wrote her an email, and she responded very quickly, highly interested to work with us. She provides a so-called plasma apheresis, which takes the lipids out of blood. And for this, there is no other technology to provide a highly sophisticated analytical platform than NMR metabolomics, because we see the lipoproteins in very great detail, more than with any technology. So we have different patient cohorts. We started with a cohort of ICU intensive care unit patients. This included a number of COVID patients, not very many because we didn't have so many in the area, and we compared those with cardiogenic shock patients, and we saw a unique signature, also a unique signature over time. There are some very interesting patients in this cohort. One, for example, was a French patient who came under chloroquine treatment with very low lipoproteins. And it happens to be that chloroquinone reduces the LDL count, and that's been published. So we found this very interesting because his LDL related particles, which are relevant for COVID-19, increase during time. So we have a number of patients who show us a very interesting signature. The number of patients isn't very large, but it allowed us a longitudinal study because samples were collected over 1 week, twice a day. We have a second cohort here on campus, which is internally called the ELISA cohort. This involves many colleagues here in Lübeck, where 3,000 patients came in over several months, giving samples every 6 weeks, and about 50 of those became corona positive. And this is probably the only collection in the world I know of at the moment where we have people before and after becoming positive for corona. And we see very nicely a difference between those who become corona positive and don't get severe disease, and those intensive care unit patients who have a severe form of COVID-19. They clearly show the high LDL, VLDL, triglycerides and cholesterol. We don't see those for the other healthy cohort. There's one more cohort we're looking at the moment. These are volunteers before and after vaccination, and there we see profound differences between AstraZeneca and BioNTech, where AstraZeneca gives us an increase in HDL cholesterol particles. This is usually seen as the healthy cholesterol, whereas BioNTech doesn't do that. This is clearly highly interesting that these two different drugs give such a different profile. So we also work with a clinic in Mülheim that carries out plasma apheresis, where we have samples before and after plasma apheresis. This is quickly becoming the probably best treatment for long COVID. With this, doctors practically remove these bad lipoparticles from blood. This is a kind of dialysis which removes lipoparticles. The patients see an immense improvement, and we use our NMR technology to follow how the lipoprotein profile changes. There we have just received samples which we will analyze in the coming weeks. At this point, we know little about long COVID, but we have a project with DRK, Deutsches Rotes Kreuz Hospital, where we collect samples from long COVID patients. I can't give you a definitive result on this yet, but all I can say is that we have an ongoing collection and a funded program that allows us to look at those over a longer time frame in an ongoing collection. So this is a highly interesting question, what goes wrong in the metabolism of COVID-19 patients. If you ask different people, you will get a different response. I personally believe that the signature that we see with this NMR technology is a signature that tells you about the propensity to develop severe COVID-19. I'm pretty sure that patients who develop a severe COVID-19 require a certain lipoprofile, which is required for the virus to dock on these ACE2 receptors, which is cholesterol dependent, which has actually been shown. So I personally think what we're looking at is a very specific profile that can tell us whether somebody is likely to get a disease. And most likely, this is also a profile that can predict whether there's a high long COVID risk. The signature that we observe clearly depends on the cholesterol in certain lipoparticles, but also on the triglycerides. I think these are the most significant contributions to this signature: cholesterol and triglycerides and LDL and VLDL particles. And the currently established medical analysis for this does not uncover this detail.